Good afternoon. Can you see my screen okay and you can hear me okay?
Okay.
Great, thank you. So I'm really looking forward to giving you today's presentation with File Korea. My name is Edward, as Heesung has told you, and I'm here today to talk to you about the Creoptix wave system. So this is molecular interaction analysis using grating-coupled interferometry. And today we're going to talk about label-free kinetic interaction analysis. On today's agenda, we'll talk a little bit about the Creoptix wave system overview, the wave technology and how it differs from SPR, surface plasmon resonance. We will also spend some time talking about waveRAPID and also highlighting our wave control software. We will then have some discussions on the applications of protein-protein interactions, small molecule, membrane proteins, and peptide interactions, all whilst thinking about kinetic parameters in a drug discovery context. So a little bit about Creoptix. We enable life scientists to accelerate drug discovery. Our headquarters is in Wädenswil in Switzerland, founded in 2009. And we are a life science tools company focused on the drug discovery market. We create our own label-free optical biosensor instrumentation based on patented grating-coupled interferometry. And we have 25 plus employees based in Switzerland, Germany, the UK, US, and Sweden. And we also work with our partners in Asia through distribution channels such as File Korea. Today, I'd like to put the presentation in context that in drug discovery, getting the binding data can be a bottleneck. So we would say that the time to data can take too long. It can take weeks to get the binding data. Existing solutions sacrifice sensitivity as throughput increases. We need expert knowledge to interpret data for decision making. This can be too slow. And we would also say that you have limited information with single data points. Finally, one point that I would like to make is that there is a large bottleneck in the drug discovery process where you cannot run crude samples in many instruments as they may clog. So our solution to this is the Creoptix wave system. It's comprised of four main pillars. We have our wave core, which houses the patented grating-coupled interferometry technology, which we will talk about today. And this gives us superior sensitivity, up to 10 per second on the off rates. Our wave chips is a second pillar, and this is an extremely important part of our product. These chips are microfluidic cartridges that are fully disposable, so we contain no internal fluidics in the instrument itself. The chip is set up in parallel with four sensing surfaces, which allows you to do some very creative experimental design. We talk a little bit more about our chips in the next slides. Pillar 3 is our wave control software, and this is really intuitive software for instrument control and data evaluation. And this contains waveRAPID and direct kinetics. This allows us to do easy data interpretation using AI. As with all of our products, they're developed in-house in Zurich in Switzerland. And finally, we have the fourth pillar, which is the wave care. And this is the after-sale support where we make sure the instrument is happy, but also that you can access the applications team through our headquarters, but also through File Korea. This is an overview of the wave system, which we'll talk about today. Here we have our wave core, which houses our innovative microfluidic cartridge where we have no clogging, fast transition, and compatible with quite harsh solvents. We have our temperature-controlled wave sampler, and we also have our automated and intuitive wave control software. How we really position the instrument within the market is occupying this top right space in this graph here. So we have superior sensitivity in the market, but we also have the ability to run very complex samples. And when we talk about complex samples, they could be crude analyte preparations where you have unpurified compounds, but they can also be crude membrane preps or human serum or plasma in the buffer. So on this slide, we'd like to highlight the high sensitivity, our robust microfluidics, and our intelligent software. So we really occupy this unique space in this plot. So now it's time to talk a little bit about the wave technology and how it differs from surface plasmon resonance. In surface-based interaction analysis, when we are looking at label-free interactions, it's comprised of many components, where we have a baseline. On our sensor surface, we'll have perhaps a protein or an antibody immobilized. We then have an association of an analyte, and we rinse the analyte off during a dissociation phase. This is done in a series of increasing concentrations, which we can then calculate the kinetic rate constants. So some other label-free biosensor technologies on the market are SPR, BLI, so biolayer interferometry, and grating-coupled interferometry. And these really focus on molecular interactions. These optical biosensing technologies measure changes in refractive index to a sensor surface. However, they are completely different technologies. While SPR detection is based on a localized field, and BLI is based on diffusion-limited conditions due to the absence of microfluidics, GCI has a much larger sensing area, which means it can see more interactions. This allows for more binding events to contribute to the overall signal, giving grating-coupled interferometry, or GCI, an intrinsically higher primary sensitivity for label-free interaction analysis. But let me explain to you how GCI works. So what I would like to show you here on this slide is comparing it to SPR, and we would say that GCI has a larger sensing area, which means it can see more interactions. GCI doesn't make use of an evanescent wave, but rather interferometry, and is therefore not as susceptible to decay as you move outward from the sensing surface. With surface plasmon resonance, you have a gold sensing surface, you have these evanescent waveforms, and upon ligand binding, we get a refractive index change. This is a localized field with a small signal. With waveguide grating-coupled interferometry, we have our waveguide here, which acts almost like an optic fiber, where you have the wave traveling across the surface in its entirety, so you have a much higher signal. This is also a refractive-based index change detection principle, so we have the output in a very similar form as a sensorgram. So in this next slide, I will show you a video which explains in a little more detail our detection principle. So this is how we see the light. Here we have our active waveform hitting our first grating. It travels across the waveguide. We can see a protein like an antibody immobilized here. We then have our second reference wave coming into the second grating, and this is in the same phase. Upon analyte binding, we have a phase shift, and it's this phase shift which allows us to detect the binding. So we're really looking at an interference pattern between the active and the reference waveform. This will then give us the output in the form of a sensorgram. So what does sensitivity mean when we talk about having superior sensitivity? Here we like to talk about signal to noise ratio with low Ormaxes. In this slide, we're showing full kinetics resolved well below 1 picogram per millimeter squared. And this means we have no need to use mass transport limited models. What I would like to highlight on this slide is, for those of you who might be familiar with SPR systems, on our y-axis, we have picogram per millimeter squared, which is the very same to a response unit on other systems. In this example, we have a target carbonic anhydrase, and we're looking at a small molecule drug, acetazolamide binding. In this first graph, we have a very high Ormax, which means we immobilize a lot of protein target onto the sensor surface. If we immobilize much less and we zoom up on this sensorgram, we can very clearly see that we can resolve these kinetics. Here we have our model data in black and our measured data in red. And what this allows us to do is to solve the kinetic parameters with much more accuracy. We also don't need to include these mass limit transportation variables as we solve the differential equation. So this could be an example also very important if you're interested in small molecule screening or fragment screening. You can have a protein on the surface that has limited activity, but we can still see the detection of the analyte binding. I'd like to talk a little bit about our innovative fluidics, means no missed data. We have very fast transition times, we have parallel flow in between the channels, and we have no integrated valves. So this is the wave chip and what it looks like. It's maybe the size of a USB stick. And here we have 4 parallel flow channels. And it's really in the design of this chip that allows us to get superior sensitivity and very fast transition times. So once again, this chip is no clog fluidics. So this contains all the internal components. So you have nothing in the instrument that can clog or break down. On this graph, what we're showing is a very fast transition time at 400 microliters per minute. Here we have a very steady baseline, and then we immediately come to the association phase. We do this through a very clever way of sample preparation. And in the next slide, I'll show you a video of how our fluidics work. So you can also see this on YouTube after the seminar, but here we look at a cross-section of the chip where we have a baseline measurement. Our sample prep is done by opening one waste port, closing the other. This is the real trick here, having 2 waste ports. We then close this one for the association phase, and it hits the sensing area with a very fast transition. So by cleverly opening and closing these ports rapidly, we can address the system quite quickly, allowing us to get these fast transitions. So I realize this was a little bit fast, so I can play this one once more. Excuse me. Okay, here we go. So once more, this is a cross-section of one of the channels. We have our sample prep happening by injecting the sample, opening this waste, keeping this one closed, and this prepares it before hitting the sensing region. We then have our association phase by opening the second waste channel. We then rapidly switch these ports and have a running buffer run in the opposite direction. So that's how we get our association and dissociation phases. What this allows us to do with these very fast transitions is not to miss weak binders. Here we have the same tool system we talked about some moments ago, but with a much smaller analyte this time. We're looking at carbonic anhydrase and methyl sulfonamide. And if you really zoom up on this curvature here, we can resolve the full kinetics. Normally, this would be described as a square pulse in the label-free community, where you would only be able to resolve the KD, or the affinity of the interaction. But here we can rank compounds and understand the association and dissociation rates much better. So I'd like to spend some moments talking about waveRAPID and our wave control software. Previously, we spoke about traditional kinetics, where we're probing the interaction by injections of varying concentration with the same duration of time. So here we can see an increasing concentration, and we have the same time for each injection. What this looks like on the instrument is a series of injections over, in this case, 900 seconds that increase in concentration. So you have all of this done in maybe 900 seconds. We then do some post-processing after it has been acquired, and we align everything to the same start point, the same rinse point, and apply a kinetic model to that. And we can solve this to understand our kinetic rate constants. So waveRAPID technology works in a very similar way, and it stands for repeated analyte pulses of increasing duration. And here the interaction is probed by injections of varying duration and from a single well, therefore a single concentration. This is how such a measurement typically looks like, including the evaluation in black. So here we're now plotting the measurement in real-time axis, which looks the same, only we have one cycle here that's being pulsed in. So we can visualize the concentration of the injected analyte at the sensor surface. And as you can see from the single well is injected is repeated in pulses of increasing duration. So this allows us to get a concentration dependency on the chip. In order to determine the kinetic rate constants, the on and the off rates, both the measurement curves here we can see in blue, and we solve the concentration function using a DMSO calibration, and we now solve this differential equation in the very same way. So we're now, instead of doing multiple injections, we're pulsing this in increasing duration. Our sensogram looks slightly different, but we solve it in the very same way. So we're probing the interaction by injections of varying duration. So this means that for one interaction, it's on the scale of 50 seconds, and we have the entire kinetic rate constant. So the affinity, the association rate, and the dissociation rate. What this allows us to do is get more insight faster. We have the same data quality with much less effort. And what we can now do is start thinking about kinetic screening, where in this plot where we show the association and the dissociation rates plotted against each other, we can see that we have interactions with very similar affinity, or KDs, but quite different association and dissociation rates. So what I would like to say is that this is 10 times faster than traditional kinetics. There is no more need for serial dilutions, and you save time and money on consumables. But why then are kinetics important? And really this is about having, in drug discovery, the first candidates are often identified as hits and then optimized for more desirable characteristics until they become drugs. Ones of desirable characteristics usually have a very high affinity. This is how strongly they bind to their target, so that a drug can be administered at low dose and has an effect over several hours. However, the affinity is a function of the association over the dissociation rate. So we have here our association, and then here we have our dissociation. So as shown here, you can have a compound with the very same affinity, but quite different binding kinetics corresponding to different behavior. So a common problem that researchers face is that traditional early stage screening techniques return results indicative of affinity only, leading to too many hits showing up in the first screening stages. Hence, more stringent methods that can provide further characterization of the binding event, such as binding kinetics, can give much richer information. So perhaps by doing this kinetic screening, we can introduce the concept of kinetic analysis much earlier in the drug discovery process, and really tailoring the best binders for the application that you're interested in. So now I'd like to show you our wave control software. This is how we set up an assay. I may need to play this one twice because it's a little bit quick, but we're also very happy to start a live session at some other seminar where we can go in much more detail. But setting up a waveRAPID assay and wave control is very simple. Here are the steps. You select the Rapid Kinetic Wizard to set up the assay. You can simply copy and paste sample information, the molecular weight and concentration from an Excel sheet, and directly paste this into a plate map. You then move on to the configuration to select the appropriate affinity range of the screen, and you can include positive and negative controls and define the frequency and how to run these control samples. You can review the final assay setup before starting the experiment, and you have the flexibility to queue assays and set up multiple screens for overnight experiments. So here you can see copying and pasting in the compounds we're interested in. We then move down here to program our parameters with controls and our affinity ranges for the pulsing scheme, and we can select our controls also. And that's it. We're then ready to run the experiment. So a little bit of pipetting and it's good to go. So that's assay setup incredibly fast, I would say, in under 2 minutes to get an assay running on the system for a waveRAPID method. Of course, you would still need to immobilize your protein in some previous steps. In this next file, I want to show you once the measurement is finished, the data is analyzed in the same software using wave control, and there is literally only one mouse click involved. The results are generated by our direct kinetics engine, which I'll speak about in the next slides. And this really includes a statistical analysis on the expected error. So you get much richer information and quality control checks on your data. So what I would like to highlight here is that despite the single mouse click and ease of use, you remain in full control of the analysis, and you can change every calculation setting for each interaction separately if required. So here we can see the software itself. We have the overview of a screen here where we have many different waveRAPID samples. What you can see also is our table that produces statistical errors and all of the binding characterization and information on this. What we also have over here is our traffic light system to tell you that you have good binding data, so you can be assured that this is a real interaction. This can help you and guide you to optimize your experiment. If you have some errors here or some red or amber lights, it will tell you you need to optimize. On this slide, we talk a little bit about our direct kinetics fitting engine and how it works. So really, this is a one-click, and that's all it takes. So to facilitate less demanding analysis, the wave control software allows you to analyze the data in a simple way. Direct kinetics is AI-powered and enables rapid one-click analysis focused on statistical estimators to provide automated calculation of kinetic parameters with mathematically valid error analysis. In this way, direct kinetics allows for fast decision-making with confidence intervals to speed up candidate selection, reduced errors with automated evidence-based estimation for all data in one click to provide reproducible data. And this really reduces the risk of artifacts on the data for greater reproducibility. So we can rank the binders by statistical error and avoid user bias by saying, this curve looks really good, but we can show with statistics that it is correct and is good. So waveRAPID is really about a saving in time and costs. You have more interaction in hours and not days. We can keep projects moving by getting your binding data sooner. We're skipping the serial dilution, so just one injection. We say that we can get binding data much sooner. So waveRAPID is 10 times faster compared to traditional kinetics, and we can analyze our data very quickly. Some of the main applications in industries where we see the Creoptix wave system being used is really an interesting field with membrane proteins. We have antibody-antigen interactions, antibody-protein, protein-peptide, and we also, because of our sensitivity, have very interesting applications with fragment-based drug discovery and screening. Our industries include pharma and biotech, contract research organizations, and academia. This allows for off-rate screening, small molecule screening, and really in-depth binding characterization. So today I've prepared some applications that I think will be interesting to the Korean market. We'll start with some protein-protein interactions, We'll then talk a little bit about fragment screening and small molecules, some time spent on GPCRs and membrane proteins, and finally some words on peptides and how our customers are using these for label-free interaction analysis and being able to resolve the kinetic rate constants, the affinity for the interaction. So here what I'd like to show is the flexibility of measuring kinetics on the wave system. This is highlighting that waveRAPID enables kinetic screens at much faster speeds. Here, our surface, our sensing surface, is a PCP, which is our chip type, protein AG surface, which allows us to immobilize an IgG antibody, which is 150 kilodaltons, and our analyte is a protein antigen. Here we show multi-cycle or traditional kinetics that many of you may be familiar from seeing on SPR systems. And here we're showing our waveRAPID run. We also compare this to regeneration-free kinetics, which is also known as single-cycle kinetics within the field. And what we like to show here is that with multi-cycle kinetics, it takes 13 hours to run this interaction analysis because you need to regenerate and run blanks in between each sample. You have this very long dissociation tail. And on waveRAPID, because of this pulsed nature, we can do this much, much quicker. And we can also see that the binding kinetic rate constants and the KDs are in very similar ranges. The same is true for the regeneration-free kinetics, and we can see highly agreeable affinity ranges and kinetic constants with this. So this really highlights how waveRAPID can benefit your workflow when identifying protein-protein interactions, in this case, an antibody and antigen. We also like to show this slide that grating-coupled interferometry and waveRAPID can resolve the tightest interactions. Here, our ligand, which is the protein immobilized on the surface, is a his-tagged large protein. This is a customer sample, so I can't go into details on the exact type of protein, but what I can tell you is that it's a his capture on our nickel NTA surface. The analyte is an antibody, an IgG, and waveRAPID is 4 to 5 times faster. So GCI resolves very fast association and quite slow dissociation constants. So we have a very flexible system. We would say that we have great correlation between multi-cycle kinetics and waveRAPID. With our multi-cycle kinetic assay time, again taking 13 hours, and waveRAPID taking only 3 hours. So this really allows you to screen many more compounds, but to get your binding data much quicker, which will allow you to progress your samples. So we have many examples of customers that are very happy. This is an example with EPFL in Lausanne, where Kelvin says that GCI allowed him to get creative and do more experiments because he could use less material. The software is easy to use because it's virtually impossible to clog the instrument. It's easy for a beginner to start experimenting and learn the experiments. You can also watch the customer testimonial on YouTube. So what I'd now like to talk a little bit about is small molecules and fragment screening. Here, this was run in collaboration with Roche, and this demonstrates screening by response. So we're not actually looking at kinetics and affinity here, but rather just comparing the known standard in the industry for fragment screening, where we look at response units and plotting this curve. So what we're looking at here is a protein amine coupled on a PCH chip. We have a positive control, which was 455 Daltons, and what we're looking at is the reproducibility on the system. So here it was a fragment library of 531 fragments. We have excellent reproducibility between the replicated runs, and these hits were confirmed by orthogonal methods. If we look at waveRAPID in early drug discovery, we have a tech note on this with Idorsia Pharmaceuticals, which are based in Basel in Switzerland, and this really showcases waveRAPID applied in early drug discovery. Here we can see the output of a screen. We can see many different hits which were identified, and this was a successful benchmarking to traditional kinetic measurements on SPR. So we're comparing this using a log factor 5 on the binding hits which were observed on a SPR system. And this significantly speeds up the process. So you're very welcome to read about this technique on our collateral website. I'd also like to spend some time talking about GPCRs and membrane proteins. And here we're demonstrating the ability to measure binding of larger molecules, proteins, to crude membranes. This really pushes the boundaries of measuring binding to membrane preps, where we do not need detergent solubilization. So in this case, it was ran in collaboration with Novartis. We have binding of a nanobody to an immobilized GPCR from unpurified membrane extracts, i.e. one centrifugation step. And I'll speak about this in a few slides where we also show some highlights from TechNote 11 and how this was done. But essentially what we're doing here is taking the crude membrane, which we've spun down by one centrifugation after lysing the cells, and we're capturing this on a streptavidin surface. So we have a biotinylated protein, and what we're doing here is immobilizing it in pulsed injections so that we can really build a large amount of protein on the surface. So channel 2 is our GPCR of interest with an N-terminal AVI tag, and channel 1 is a negative control membrane with an AVI tag. And what we can resolve here is the kinetics with a nanobody, which is a single domain antibody. Here we see a good Rmax, we have our association, dissociation, and affinity for the interaction. So this is really a protein-protein interaction in a crude context. So here I'd like to talk about our TechNote 11, which is the quick characterization of binding onto unpurified GPCRs. So the standard GPCR purification process is quite long and arduous, where you have a solubilization step, an ultracentrifugation, filtration, IMAC purification, concentration, overnight cleavage, which can be sometimes optional. You have a negative purification tag removal, concentration, gel filtration, and once again another concentration. Then the product is ready for analysis. This is a large-scale process, which is normally a 2-day workflow with 2 liters of cell culture. Excuse me. Here we talk about quick and dirty GPCR purification process, where we have the solubilization step and then one benchtop centrifugation. So here we can have a small scale process, 1 hour workflow, and at 30 milliliters of cell culture required. So this allows you, and what this TechNote is showing, is binding to semi-purified membrane preps, which saves time and sometimes aggravation on purification when you have difficult to purify GPCRs. The assay details are, we have an analyte, which is a purified G protein, which is DDM detergent solubilized, and we activate the GPCR with an agonist before starting binding with the G protein. Analyte plus agonist. So we run this in a competition mode using the ABA injection on our device. And here we can understand the 2 interactions which are involved for this mini G protein coupled interaction. On this next slide, we're looking at crude virus-like protein preps. And here we can say that kinetics are possible. And what we're showing here are membrane-like, virus-like particles, nanodiscs, proteoliposomes. So these are non-purified membrane proteins in detergent. The assay details is a crude VLP, and this is captured via a lectin, which is a wheat germ agglutinin surface. And we have antibodies as the analyte. This is not a small molecule interaction, but a protein-protein. And what we're showing here is our reference surface. So we have this anti-CXCR4 antibody kinetics on an empty surface and then on an active surface. So we can resolve the full kinetics here. Here we have the demonstration of allosteric inhibitors on purified orthosteric compound GPCR complexes, and we're looking at the kinetic characterization of an allosteric inhibitor, so a small molecule. And what this demonstrates is binding to fully purified membrane protein. Now we're not talking about a PrEP, but you can see small molecule binding. These types of assays are often run on other technologies. However, on many SPR systems, it's not able to measure the same degree of fast off rates that we are. And this is due to the valve switching and the innovative fluidics that we spoke a little bit about earlier. So with other technologies, they would recommend doing an equilibrium analysis. However, in our case, we can resolve the full kinetics. The assay details here. This is a class A GPCR detergent DDM purified. We have an orthosteric compound, GPCR complex. We're immobilizing on our nickel NTA surface. The orthosteric compound is in saturation, so it's in the running buffer. And we can see that both the allosteric compounds are able to bind with similar affinities on the GPCR orthosteric ligand complex. Finally, I'd like to finish this session with a discussion on some protein-peptide interactions and how you can use the wave to understand these interactions. Here we have one of our customers, PepScan in the Netherlands, demonstrating purified peptide samples in buffer and bovine serum. The assay details here are peptides which have been selected from a combinatorial peptide library used as capture reagents for an immunoassay detection of therapeutic monoclonal antibodies. What we see here is the kinetics in a buffer, and we also see kinetics in 50% bovine serum. In this example, we're looking at plant LRO receptors with ligand peptides. This is a very nice publication from the University of Geneva, which you can read by clicking this link. It's in PNAS. And what we have here is our surface PCP wave chip, which is a quasi-planar surface, allowing you to study very large protein-protein interactions or protein peptides. Our ligand was a recombinant biotinylated plant LRO receptor, which is in the range of 70 to 90 kilodaltons. This was immobilized on the sensor surface. And what we're looking at here is an analyte peptide around 20 residues long. And we can look at all of these interactions and resolve the KDs of very tight binders, but also understand peptides with fast association rates and slow dissociation rates, and even fast on and fast off. So we have a very flexible system to study these protein peptide interactions. So here again we have an example with characterization of crude peptide samples. Here we show the successful identification of nanomolar CLIPS binders to a therapeutic monoclonal antibody used to treat autoimmune disease. So these are macrocyclic peptides which were ran by PepScan and that allowed them to really understand which of these were true hits and true binders. There's also a very good poster on this, which I would recommend you to look at. And we've already seen this slide, so I'll quickly skip on. One other little application that we have many applications that you can see on our website and in peer-reviewed literature, but what I'm showing here is the assay details is a DNA-DNA interaction, and we can look at reproducibility across very high and low ligand densities. Here our assay details are a 10 nucleotide single-stranded DNA biotinylated ligand was captured on the surface of our sensor chip via streptavidin biotin capture. the complementary 10 nano or nucleotide single-stranded DNA analyte was injected in duplicate concentrations from 4 micromolar to 0.2 nanomolar. And what we can say here is that we have excellent kinetics that fit a simple one-to-one interaction analysis. So this is the seminar I had prepared for you today. I hope that you found it interesting and useful, and please feel free to reach out to us through our great partners, File Korea, for any additional questions you may have. Thank you very much.